Good morning, everyone, and welcome back to the Agronomy and Action Insight series. We're very excited to have you with us here this morning to talk some agronomy. This is our fourth of 6 webinars that the Golden Harvest Agronomy team is putting on this winter to bring informative information for you and your operation. Today, we're going to go through the topic of the evolution of corn rootworm management. When it comes to content for the corn rootworm presentation this morning, we have one presenter with us to carry forth this message. We have Andy Heggenstaller, the head of Syngenta Agronomy, with us this morning to talk through this evolution of corn rootworm. So with that, I'm going to pass it right over to Andy so that he can get us started this morning. We appreciate you joining the webinar. Andy, over to you.
Julia, thank you so much. Good morning, everyone, and thank you so much for taking the time to join us. Really excited to share some Golden Harvest agronomy perspective on corn rootworm this morning. It's a topic that my team and I spend a lot of time on, as I'm sure many of you, if you've taken the time to join this webinar, do as well. I'm going to really do this in 3 parts this morning. We're going to go through some basics. Some of this will be review, but I want to make sure we set the table the right way. We're going to talk about management, how that's evolved, and then finally we're going to spend some time at the end of the presentation covering the current and future state of biotech traits for corn rootworm control. So obviously, if you're on this call, you're familiar with this pest, have interest with it, and have probably dueled with it in the past or currently are. It is, in fact, corn rootworm, the most destructive insect pest in U.S. corn production, variously estimated at $1 billion or more in damage. There's 2 primary species that affect us in U.S. corn production. That would be the western corn rootworm and the northern corn rootworm. And the primary damage, of course, that this pest causes is through feeding on roots, subsequent lodging, and loss of yield due to nutrient and water uptake. But we also often have issues related to pollination when adults feed on silks, and that can be an issue in some cases as well. As I mentioned, and as I think most of us know, there are 2 species of corn rootworm, but I think one of the more important things to understand at a high level is that not only are there 2 species that have slightly different but overlapping ranges, but both of these 2 species have developed a variant that has effectively allowed, in some cases where that variant populations exist, has allowed them to overcome the easiest or the primary method of corn rootworm management, and that is rotation out of corn. And again, I think probably a lot of us are familiar with this. It's been at the front of our minds more in the last couple of years, particularly with regard to the northern corn rootworm variant. But essentially, there's a variant of the western corn rootworm, ironically, that tends to be in the eastern corn belt, and that they have adapted to lay their eggs in soybeans, thus being present in corn the next year when the larvae emerge and the beetles emerge. Similarly, there's a variant of the northern corn rootworm, primarily in that Iowa, Minnesota, eastern Dakotas area, that has found a way to, or evolved to have, extended diapause, where essentially the eggs remain in the soil through the soybean phase of the rotation, thus becoming a problem again. And as many of you, if you're in the northern corn belt, probably are aware, this variant has become a greater challenge for us over the last several years. What this map here is showing us in the blue is sort of the historic primary geography of where we've seen most of the northern variant, the variant of the northern corn rootworm. And through some work that the Golden Harvest agronomy team did, it's not all encompassing, but where our agronomists have done sticky traps and looked at what's going on, those are those yellow boxes. And we can really see that the presence of the variant was very high in Iowa, Minnesota, parts of northeast Nebraska, as well as eastern South Dakota this year. And I think anyone who's working with corn rootworm management, whether you're a farmer, seed advisor, agronomist, you know that both the intensity and the frequency of this pest have increased in recent years. And so that's creating some new challenges for many of us as we're thinking about managing the northern corn rootworm, where now rotation isn't always as reliable as a management tactic as it has been. Whether we're talking about the northern or the western, the data on this slide is the 2 combined. One of the biggest first things to think about at a high level from year to year is what is likely to be the pressure in a particular region the following year. And the easiest way, it's not perfect by any means, But the easiest way we can understand this at a high level is through adult counts from sticky traps or other measurements that people have done during the peak of the prior season. And that's what the data you're looking at is here. This is from a multi-organization, several companies, universities. It's an effort that Syngenta participates, Golden Harvest participates in. And the colors kind of speak for themselves. There's a legend in the upper right. As the circle gets bigger, and the number of adult beetles found in the trap at that location increase. And what this tells us, again, it's not always perfect, but it would indicate to us that we're likely to see pressures similar next year to what we've seen in recent years. And what I mean by that is we're seeing Iowa, southern Minnesota, South Dakota being a hotbed of corn rootworm activity, both western and northern. Whereas we've really noticed in the Eastern Corn Belt, or at least I feel like I've noticed and my team's noticed over the last several years, we just haven't had as high a pressure, say, in Northern Illinois, Southern Wisconsin. Again, in this map, the data is a little bit limited in Wisconsin, so we'll see. But it does, last year's beetle emergence does tend to point toward Iowa and parts north and west, probably being the highest pressure that we're likely to see based on the adults that were flying around last summer. Now, that's a great high-level view, but for an individual farmer, agronomist, or anyone who's managing this in a specific area on a specific set of fields, a lot of that can go out the window, and what's happening locally in terms of management or factors in the local environment can ultimately drive what's really happening. Just in brief, some factors that we've really seen drive differences. Anytime we have an area where there's been beetles present in the current year, wherever we have fields that were planted before or after the typical planting date of the season, or where we have hybrids that are much earlier or later in relative maturity, those fields that are on the ends of those distributions, whether it be planting date or maturity, they tend to be places where the beetles will flock to. The early beetles will go to those early fields. The late beetles will move to the late ones. And those fields end up kind of becoming trap crops in a way, if you will. So if you've got those kind of fields in your operation, and you saw a lot of beetles last year, that would be some of the first places I'd be looking to think about what am I going to do? Because they're probably the places where my egg counts are likely the highest. Obviously, where we have continuous corn or the presence of grasses continuously, we're going to have challenges. If we've got presence of the variant, which I know many of us in Minnesota and Iowa are thinking about right now, that's obviously something we need to think about, even if we're in a corn soybean rotation. And then finally, don't forget about weed escapes or cover crops. Any grass type, whether it's a volunteer corn or a rye cover crop, all of those things can create opportunities for localized beetle egg laying and issues the next year, even if there wasn't soybeans in the field, even if there wasn't corn planted in the field the prior year. So probably a lot of that is familiar to many of you, but just wanted to call out some of the things that we see most often on a field-to-field basis within an individual farming operation being important. With that, Julia, I'd like to go to our first poll question. You just heard me talk there, everyone, for 3 or 4 minutes about what I'm seeing drive corn rootworm pressure. I'm curious on your farm or farms that you work with, how would you describe over the last 2 or 3 years the changes in corn rootworm pressure? Is it not a problem, kind of decreasing? Is it something you need to manage, but it hasn't really been changing? Or is it something that you see as an increasing challenge, or a variant has changed the game for you and that's making it more difficult? Kind of low, typical, or high challenge level is the question. Wow, we had a pretty big breakout there. About a third of you said not a problem or decreasing. Our winner was at about 42% is I need to manage it. The pressure's steady. And about a third or a little less than a third of us did say that it's an increasing challenge. So we have a good mix of folks in here. I'll try to, in my next session, which will probably be the most interesting from a management perspective, I'll try to talk about how we think about management in those different buckets, and what different tactics might be best applied. So with that, let's talk management. I think the two big ideas, and then we'll get into specifics and we'll talk about specific actions, but at a big level, sort of conceptually, the two ideas that I've always thought are most important with corn rootworm management is number one, think multi-year. Don't think in a single year. Think over a series of years and how things will fit in that way, because this pest has a memory, a good friend of mine likes to say, and what happened last year is almost certainly going to relate to what happens this year. And then the other piece which fits well with that is think about multiple control tactics and how you deploy different control practices over a cycle of whether it's a crop rotation or a cycle of years in your farming operation. This graphic, again, it's meant to be more conceptual than actionable. We'll get to the actionable stuff in a second. But the idea here is how do we match crop rotation, insecticides, adult control, different trait packages, and how do we bring all those things together, whether we do or don't have rotation in there, and that ultimately becomes one of the biggest drivers in how we make our decision. I really like this quadrant graph here that breaks things into kind of, do we have no or low corn rootworm pressure? Do we have rotation or no rotation? Because I really think the way I think about or recommend to people to think about of the tactics that are on the table, they're going to depend on which of these buckets I'm in. And I think the first one kind of goes without saying. If I'm in a steady corn soybean rotation, or maybe even a longer rotation than that, and I haven't had challenges with corn rootworm, I'm probably not that interested in what's happening on Andy's presentation here today. If I have concerns that the variant or there might be something going on locally, maybe I'm going to do some monitoring, or maybe I'm going to consider some traits. But probably if I'm in that bucket, I'm just in that group. It's not a problem for me. I think the bucket that I see increasing the fastest, particularly in that kind of Iowa to the north and west, is this area where I am in crop rotation. There has historically been corn rootworm pressure in my area, but my crop rotation has enabled me to manage that with minimal other actions required or expenses or inputs. And that's where we're starting to see that change, particularly with the growth in the frequency and incidence of our northern variant. We're seeing rotated ground where we're going to begin likely to think about some additional practices. And the two that would come to the top of the radar for me in terms of their effectiveness versus cost and complexity would be corn rootworm traits and high rate insecticide seed treatments. And I'll talk about both of those in a minute. We may also in this situation consider some soil applied insecticide, and we may be thinking about doing some monitoring to understand if a particular field actually had the variant in it that year. If we are not in a crop rotation, so we're thinking now kind of that continuous corn world, if we have no or low corn rootworm pressure, we're probably doing traits. We may want to consider doing a high insecticide seed treatment. And in my experience, the farmers I've seen manage that continuous corn world the best, even if in any given year the pressure is not through the roof, they're doing something to monitor it. And they're keeping an eye on how those populations are changing year over year. We'll talk about sticky traps in a minute, but there's more sophisticated technology available today with DNA sampling of soil, where we can actually estimate egg counts in the soil by taking soil samples. But as soon as we get to continuous corn, I believe that monitoring becomes something we want to get pretty serious about in some way or another. And finally, the apocalypse scenario where we're in long-term continuous corn and we have ongoing high pressure and or variance. Essentially, at this point, everything comes onto the table. The only one that might not be is whether you're going to actually take an action to control adults. We'll talk about that in a little bit. That's one where we've seen kind of variable results. I know some managers swear by it and have had really good success. Others, I think, have seen mixed results. But the fact of the matter is all of our tools are going to come onto the table at that point. Doesn't mean we use every single one of them on every acre every year, but we're going to be needing to think about all of them. So I want to show some examples. That's maybe a good heuristic or a good kind of rule of thumb there, but let's look at some actual examples and look at some data of what the Golden Harvest Agronomy Research Team has seen with some of these combined practices. So this first one that I'll look at here is where we looked at how do we see across a range of corn rootworm pressure on this graph as we move from the left to the right. The pressure, the incidence, the number of eggs, larvae, adults, however you want to frame it up, is increasing. And we're measuring that based on root feeding, anywhere from no nodes of root feeding the whole way up to 3 complete nodes fed off. By the time we get to 3 nodes fed off of a corn plant, it is almost certainly laying on the ground, and it's a disaster. So in these studies, we try to capture a range of pressures to see which types of things are going to fit in those different levels of pressure. That heavy black line there, that's kind of the control line. That's showing the level of corn rootworm feeding as measured by nodes at the half node interval when we had no trait and no insecticide, soil applied insecticide involved. That's what happens if we just didn't manage it. And you can see we had the whole way up anywhere from just under a node of feeding with no control, the whole way to 2.7 nodes. So we got a nice range of pressure. And then those other colored lines correspond to the legend there at the lower left. That gray line is showing us the reduction in feeding when we had no trait, but we used 4,6,5G soil applied insecticide. And then the blue line that jumps down quite a bit, that's across all those environments where we didn't use an insecticide, but we used hybrids with the Duracade trait stack. And then finally, the orange line is where we actually used both soil applied insecticide and Duracade. The point, I believe, of this research, and we've seen this play out very similarly in other studies we've done, is when the pressure gets really high, there is absolutely a value of stacking a trait with an insecticide. When we're in that kind of one, maybe 1.5 nodes of feeding, we can usually get by with the trait alone. Once we get beyond and we're going into that 1 3/4, 2 or more nodes of feeding, usually we're going to be unsatisfied with the trait alone. We'll talk more in a minute about how traits control insects, but it's not because the trait's not working. It's absolutely working. And this data shows it. That trait is reducing compared to the black line. It's reducing the node feeding significantly across all pressure levels. The thing is, once we get above a half of node of feeding, we notice it. We start to see yield impacts. We start to see some corn lodge. And you notice there when we cross about that 2 nodes of feeding threshold, the trait alone is struggling to keep us below that threshold of 0.5 nodes of feeding. Another tactic that I've become increasingly interested in, and I'm seeing more and more seed companies offer as an option, Golden Harvest absolutely does, and I'm seeing more and more managers look at as an option. I just haven't until recently seen a lot of data on it. And so Bruce Battles and his team, if you had a chance to join the webinar last week, they talked about a lot of the different work that they do here at Golden Harvest. And one of the studies they did a year or two ago was look at how did a high rate Cruiser Max Vibrance seed treatment, that's comparing that 1250 rate to a standard 500 rate. How did that affect corn rootworm damage? And so it's a very similar graph, everyone. That black line is the control. We've got no insecticide and we've got no trait. Very similar pressure level range. In fact, this was in that same study, so it's in the same locations as that data I just showed you. And we notice here that in the absence of any trait at all, that high rate insecticide seed treatment did buy us some additional control across most levels of pressure. As the pressure went up, the additional control, that's the difference between the blue and the black line, it increased. But alone, if we had any significant pressure, the high rate insecticide seed treatment alone was not going to provide enough control. And that probably makes sense, right? We don't expect that just a seed treatment is going to keep a moderate to high level corn rootworm population in check. The orange line there is when we just had Duracade across those pressure levels, and that gray line is when we combine the two together. And the point here is we notice that we have that incremental effect, that additive effect. If the pressure gets bad, if we combine Duracade with a seed treatment, we can buy ourselves some additional control. As I'll talk about in a minute, the way these traits work, they tend to be expressing more middle to later in the season. So any insecticide tactic, whether it be a seed treatment or an in-furrow application, they really tend to help us because they provide control earlier in that kind of June timeframe, where we're not getting as much activity from the trait because the roots of the corn have not yet fully developed. One last thing I would say here, and I think it's obvious from the data, a high rate insecticide seed treatment is not going to provide us the same level of control as an in-furrow force type application. I think that probably goes without saying. This is, although a simpler and lower cost way, and if our pressures are more in that moderate level, it might be a nice combination with our Duracade trait. And additionally, that high rate insecticide seed treatment is probably going to provide us some other benefits beyond corn rootworm control. I mentioned this a couple of times, so I won't belabor it too much more, but I'm a big fan, as is my team, of taking some time to monitor what's going on with adults. I really think there's 2 big reasons why this can be valuable. It's not going to work or be done by everyone, I understand, but we can do two things with it. One, we can get an idea of what we think is likely to happen next year. So that helps us in that multi-year thought process with corn rootworm. And then probably the more immediately actionable thing, and I've seen people use this to great success if they're diligent about it, is we can put these sticky traps out and every week, put a couple in each field and every week remove them. And when we see the number of beetles on that trap peak out and kind of cross over that 20 or more over a week, we're probably approaching the peak emergence. And that can be a really good time to time an insecticide application for adult control, which would again help us for our issues for next year. I've seen a lot of different protocols out there. Here's the example of the one the Golden Harvest Agronomy team uses with our sales team and our seed advisors. To really do it well, you need to put a half dozen or more traps in a field and check them pretty regularly. And I encourage that if you really want to get precise. But I also think there's a lot of value of doing what I would call a sentinel trap and just having one or two traps on the edge of different fields and kind of getting an idea across your farm. You're not really trying to estimate the number. You're trying to really understand what the emergence distribution looks like. And I think that works particularly well if I'm trying to time an insecticide application on the farm. All right, I would like to spend the last part of our time together here today and talk a little bit about corn rootworm biotech traits. Obviously, this is our dominant and has been for almost 20 years, our dominant technology to control corn rootworm. It's fundamentally changed how we think about corn rootworm as a pest in the US. And I would argue that a lot of the way we farm corn in the US today in the core central and northern corn belt has evolved as a response to the presence of these traits. One of the places that I really like to start is thinking about how a trait affects and ultimately kills a corn rootworm larvae, because it's very different than how an insecticide works. When a corn rootworm larvae comes in contact with a force insecticide or that seed treatment, they're going to die within hours of contact with that, typically hours to a day of contact with that toxin. Our BT traits and our corn rootworm traits don't work that way. We couldn't put that level of toxicity into a corn plant. It wouldn't work well, right? So they're expressed in this crystalline protein. Typically, we see some technologies coming now that work a little different, but most of what we have used and what we're using to control corn rootworm today still relies on these cry proteins. If you've heard cry, it refers to crystal. It's like the reproductive state, if you will, of this bacteria. Most of these, or all of them to date, come from a bacteria called Bacillus thuringiensis. When that crystal protein, which is present in the cells of the roots of the corn plant, when those larvae eat those roots, consume that crystal, the crystal dissolves in the gut, in the foregut of the larvae, and then a toxin is released out of that crystal. That toxin binds with the cell wall linings in the midgut of the larvae, and it essentially cuts open the cells in the stomach lining of the larvae. And essentially at that point, the larvae stop feeding. They don't die immediately, but they do stop shortly after consuming roots that have the toxin crystals in them. They stop eating additional roots. They get sick, essentially. Imagine if you or I ate a bunch of glass, we'd probably not be hungry for a little while. What happens is most of them ultimately then desiccate and die in the soil. They stop feeding shortly, but a little bit more, or depending on the technology, a lot bit more time goes by and the larvae dies. What can happen as a result of this, though, sometimes the larvae will stop feeding, it will get better, it will continue feeding, and it will be able to survive through. This is both the reason why the traits have worked so well for us and why we see resistance develop to them. Is they're very effective, but they're compared to an insecticide, they're what we would refer to as a low dose technology. And that low dose nature allows us to deploy them through a plant, but it also results in a situation where some larvae over time are able to select for and those that survive pass on their genes, and that's how we see resistance develop in populations of corn rootworm. I talked about the adaptation of the northern and western variant. That really happened in the late 20th century, say 1980s and 1990s. That began to see something that became evident, that corn rootworm were finding ways around rotation. And it was within 6 years of the introduction of the very first corn rootworm trait that we saw the first population develop resistance to that. And over the subsequent 20 years since the introduction of the first corn rootworm trait, we've seen either field or laboratory examples of resistance or at least increased survivability on populations exposed to the same traits over time. I'll show you data in a minute. I don't want to mince words or confuse anyone here. Every single corn rootworm trait in the industry today still has utility. I absolutely believe that, and I've looked at a lot of data that makes me believe that. But at the same time, in any given field or different areas, we are seeing an increasing number of populations develop tolerance, in some cases cross resistance. One larvae that's population that's been exposed to one trait has increased survivability on another because all of the BT traits have some similarity. And so as a result of this, as an industry, to keep this really valuable and frankly high farmer ROI technology available, we continue to need to invest and develop new ones because as time goes by, the broad adaptation of the older ones becomes more limited. We see this very similar to how we think about herbicide traits in soybeans today, right? So I'm going to do my second poll, and I'm not trying to trick anybody here. I just think it makes a great segue. How many unique biotech corn rootworm control proteins are available in the U.S. market today, seed corn market? 3, 5, or 7? What do we got, Julia? Hey, nice job. 55% of the group got the question correct at 5. That is, in fact, the correct answer. If you answered 7, only 4% of you answered that, but you're actually right. That's an answer to a slightly different question than we'll get to in a minute or two. But there are 5 unique proteins available today. That very first one I referenced a minute ago was introduced in 2003, that from Monsanto at the time, Yield Guard rootworm. And 21 years later, 2 years ago now, Bayer now released the most recent one with Smart Stacks Pro. In between that time, Herculex rootworm from Dow, DuPont, now Corteva, and then 2 traits in that middle area as well from Syngenta, Agrosure rootworm and Agrosure Duracade. You can see on the right, I just got this data recently from some of our strategy folks. You can see the number of acres in the United States Corn Belt that have this trait on them and the percent of those acres that represents in terms of corn rootworm acres. And so we can really see that we have 3 traits, Yield Guard rootworm, Herculex rootworm, and Agrosure rootworm that are making up the majority of those acres. We can see that Smart Stacks Pro is making a run and starting to grow, and I anticipate we'll see that newest protein in various stacks begin to proliferate over the coming year. If you answered 7, you were also right, but that was the answer to the question of how many combinations of these different traits are delivered in a branded stack by one company or another. So there's 5 unique proteins, 4 of them are BTs, and this newest one, which we'll talk about in a minute, from Bayer, this Smart Stacks Pro, or this RNAi, as you've probably heard it referred to, that is a different mode of action from the BTs. So that's something significant. It's the first non-BT corn rootworm biotech mechanism we've seen. But again, to the point, these 5 different proteins are available in different combination or stacks to bring different modes of action of control from different companies. And you can see that Bayer, Corteva, and Syngenta, all of us in some way, shape, or form are accessing proteins from one another to create different combinations and technology offerings for farmers. So we can have those multiple control mechanisms. I won't go through all of this. I realize there's a lot here. I wasn't intending everyone to memorize or pick up all those different CRW protein codes over there. But I think one thing that's really important about this is 2 of these offerings are essentially brand new. And so we'll talk about that in a minute, but there's 2 of these 3 offerings with the RNAi that have been effectively in no farmers' fields, little or no farmers' fields to this point, and we'll start to see them in the marketplace over the coming year. I want to step back from the new stuff for a minute because this is a question that I get a lot or I hear people talk about a lot. And that is, are some of these traits better than others? And the answer to that is mostly no, but it can depend on the history of a given field. So last time I'll show you one of these charts. This is, again, one of the studies where we've looked across a range of fields. that have different levels of corn rootworm pressure as measured by how much root known feeding we have in the absence of management. And what you're looking at here across a bunch of different hybrids is the level of control provided by hybrids, including the Duracade trait stack from Syngenta Golden Harvest, the Chrome trait stack from Corteva, in this case in Pioneer hybrids, and the Smart stacks. This is not the RNAi. We're evaluating that for the first time this year, we evaluated that for the first time last year. But the gray is Smart stacks containing hybrids, in this case in a decal bag. And what we noticed across a lot of locations, a lot of years, and a lot of level of pressures, that again, that black line is untraded hybrids, the blue line's Duracade, the orange line's Chrome, the gray line is Smart stacks. And we see that they're way more similar to one another. They all provide very high level of control, especially if we're below that 1.5 nodes of ambient pressure, if you will. I have been in fields in my career where I've seen one of these traits have serious issues and another one look really good. I've seen all manner of combinations of that before. And so a lot of times in a given field, if we have a split planter or we have a plot or we see one particular trait stand out better, or a hybrid with one particular trait stand out in a good or bad way from others, oftentimes that can be due to either what historical traits have been used in the field, what resistance pressure is on the field. And more often than that, it can oftentimes be determined by differences in the hybrids, in the genetics in the field, and not actually the traits themselves. Here's an example that I really like from one of our agronomists in Minnesota several years ago now. This was a very, very high pressure field. You can see the roots on the right. You can see the root rating at the top. That was with a couple hybrids that were just our AgriSure above trait stack. They didn't have any rootworm control traits at all. 2.4 nodes of feeding. That's what that looks like there on the right. It's a bad day. And we had a couple hybrids each in Smart Stacks, Duracade, and Chrome in that field. And you notice in that field that all three of those hybrids provided a very similar level of control. And in all three of those cases, it was not a level of control that you would have been happy with. 1.5, 1.6, 1.8 nodes of feeding is not a good day. It's better than 2.4. The point I wanted to make is in this example, which is one of the more extremes I've seen in an actual farmer full-scale field. This is not a plot, guys. This is a full field with a bunch of hybrids stripped in it on a bad rootworm day. All three of these stacks looked pretty similar, and all three of them were challenged compared to like the level of pressure that was out there. You will notice there's some differences in yields, and that's really driven more by the genetics than it is by the trait itself. One last point on this genetics versus trait. I just think it's important to think about when we have rootworm challenges. This is just with two of our own Golden Harvest hybrids here in a large plot in northern Iowa from a couple of years ago where we had very high pressure. And there were hybrids from a number of different trait packages across the industry in this field. And we could, if we just looked at that 03B96D hybrid, we might draw the conclusion that Duracade is not working. The reality is there were a lot of other hybrids in that field that had Duracade in them that looked as good as anything else did under that level of pressure. And so again, my point is think about the genetics you're planning as well as the trait if you're going to be dealing with some rootworm pressure. I've seen differences in the root scores in hybrids have as much impact on how a rootworm situation is handled as the trait differences in the field might be. And my advice is to try to pick those hybrids with better root scores, if nothing else, when we're looking at those fields with the heaviest pressure, because that'll combine with the trait to put us in the best position possible. Let's conclude, talk a little bit about the future. I referenced on that slide a minute or two ago that there's some new stuff coming. Most of it is actually not new technology, with the exception of the RNAi. It's different combinations of combining that RNAi with existing things. So I'm not here to represent Bayer or Corteva, but I do have visibility to what's going on, and I'll just share what they've announced publicly and what they're clearly doing. And then I'll conclude by giving a view of what Golden Harvest is doing and what Syngenta is looking forward to in this space. So as I mentioned earlier, the Smart Stacks Pro hybrids were launched in 2022, and we'll start to see them be pretty widely available in the market. They started to be last year. We'll see even more of that this year. They're effectively combining that new RNAi with the original Yield Guard rootworm trait and the original Herculex rootworm trait from Corteva. I mentioned some new introductions. Bayer had a little bit last year. We'll see more of it this year for a really 2025 full launch of a new technology called VT4 Pro. And interestingly enough, this is not a new technology that combines a new trait. It's a version that Bayer is going to offer that'll include only their RNAi and their Yield Guard rootworm. It will not include the Herculex rootworm stack from Corteva. I could speculate what their strategy is here. I'm not going to do that. I don't think it's my place to do that. But I will say that this will be a unique offering that will combine their two proprietary traits, and they won't have any in-licensed traits in it. Corteva has obviously been focused on Chrome. I think that's been fairly successful for them. That's a combination of their Herculex rootworm trait in a new molecular stack that's caused them to have some benefits in their breeding process. But that also includes the Agrosure rootworm trait from Syngenta. And so that will be their dominant trait in the marketplace for several years to come. They are introducing this year. I don't think we'll see significant volume of it until 25, 26. But Corteva is introducing a new three-way trait stack that will include their Chrome version of Herculex, the Yield Guard rootworm trait from Bayer, and the RNAi trait from Bayer. So they're going to be looking at two BT traits and an RNAi. That'll be, as we get to the end of the decade, second half of the 2020s, that will become their dominant rootworm stack in the marketplace, offering them three modes of action across those two families. So what about us? We're clearly, and have been since 2017, 2018, focused on our Duracade stack that combines our Agrosure rootworm trait with our E-Cry Duracade trait. But we are going to be making a step forward as well in the next couple of years. We are going to offer, which I'll talk about in more detail on the next couple slides, we're going to be offering an as yet to be named trait stack. We call that internally MSRW for multi-stack rootworm. We anticipate to launch this the year after next. It is in our late stage testing for the first time this year in a number of different hybrids. It includes a molecular stack of our existing rootworm traits, along with essentially the Chrome version of the Corteva Herculex rootworm trait, making it the first rootworm control stack that has three unique BT modes of action in there. And so it's not just three BT modes of action. That in of itself would be nice. But what makes it, I think, more compelling is that both of the invents, if you will, that are introduced into the corn plant to provide these three traits, both of them themselves are what are called a molecular stack. What that means is, in the case of Agrosure rootworm and Duracade, we've taken the events, the proteins, the genes that create the proteins for those two traits, and we created one single new trait out of them. This is very similar to what Corteva did when they combined the Herculex 1 and the Herculex rootworm traits into the Chrome event, what I referenced as DP4114 on the last slide. The benefit to Pioneer, to Corteva of that, and the benefit to Syngenta of doing this is it makes trait integration and breeding simpler, which means you put new hybrids out faster, which means you get more yield faster in rootworm traits. So we're really excited not only to bring three different rootworm control technologies together, but to do so in a way that we have two molecular stacks, which we believe will allow us not only to have highly effective rootworm control, but to do it fast and do it in hybrids that bring a lot of yield and agronomics to the market as well. We did our first testing with it this year. Not everybody loves it when I show this graph, but I did want to show it because I think it's cool. And this is just a study across two locations. We'll do a lot more of this over the next couple of years. We put in here an untraded hybrid, as we often do, kind of as the control check. That's that blue bar. And you can see in these locations, on average, we had 1.5 nodes of feeding. So there was pressure. There was real rootworm pressure out there in these fields. And those green and yellow and black bars to the right, those are single hybrids that are commercially available from either us, Pioneer, or DeKalb or today in the Duracade Chrome Smart Stacks and Smart Stacks Pro flavors. Those two red bars are two experimental hybrids of ours that have our new MSRW stack in them. I've labeled them as hybrid 1 and hybrid 2. I doubt we'll commercialize either one of those. Those are sort of experimental hybrids, so we can begin to look at how the trait works in commercial germplasm, or germplasm that would be like what we would commercialize. And what you can see here is, again, all of these traits, as I said before, they're all providing a very high level of control. When you're below that 0.5 line, that's control. And what we notice is our multi-stack is providing benefits. They're small, but it is showing us under this level of pressure benefits versus a number of different stacks out there. And that gives us encouragement that this is going to provide value in the marketplace over the next 5 to 8 or more years from the time we launch it. Super excited to share more with everyone about this. We can anticipate a name for this coming this fall, or at the very latest, the very beginning of 2025. And so we won't have to call it MSRW. We'll have an actual trait brand name for it. Picture's worth 1,000 words. This is from our research facility in central Iowa, showing you 3 different hybrids. These are hybrids from plots that were in that study I just showed you. Just again, this was an extreme example. That's not 1.5 nodes of feeding. That was the average for the study. This is like off the scale. This is 3 nodes of feeding here. And you can see in that level of pressure, here's hybrids with 2 of the existing stacks and our new stack showing just looking really strong. And the new MSRW stack in particular looking really strong under that level of pressure. So I'm going to wrap up and see if we've got any questions, Julia. I'll just make a couple concluding comments here this morning, everyone. I think you hopefully picked this up, but I encourage everyone to think about corn rootworm over multiple years. If it is something that's a challenge for you, think about your rotation as well as the tactics that you're going to use in a single year. I would also say that in most cases, there will be exceptions, but in most cases, these dominant 2 mode of action stacks, this would be our smart stacks, our Chrome, our Duracade, we see time and time again in larger scale studies that across a number of fields, they provide similar levels of protection. But we're going to find individual fields where 1 or more of them are going to have challenges. And frankly, that's going to increase in the coming years. And what we see, I think, as the light at the end of the tunnel, so to speak, is we see all the players in the industry having some new offerings, more than what we've seen since the mid-2000s. And so there's going to be a lot of offerings out there for you as farmers to help manage rootworm into the future. And technology is always good for agriculture. And just finally, we'll be in that space too. And we have a new stack that we're really excited about to get into plots and to get out there with farmers here in the next couple of years. So, Julia, I will conclude there, and I'd be happy to take any question that the audience has.
Great. Thank you, Andy. Appreciate the information that you brought forward over the last 45 minutes. What a great presentation to walk through the key management and traits in the industry today and for the future, right? That's very exciting. If you have follow-up questions, please reach out to your local agronomist and or sales representative and GHX specialist. As Andy mentioned, and as you saw last week, we have a lot of great information in our agronomic research review, the Agronomy in Action research review that can answer a lot of the questions that arose today as well. So that concludes our call for this morning. We look forward to seeing you next Thursday for our 5th session in the Agronomy in Action Insight series. But for today, that concludes our call. And I want to thank Andy for all of his time here this morning. And I hope you all have a good and safe Thursday. Thank you, everyone.